Hello, tutor, and welcome everyone joining us for this deep dive.
Hello. Good to be here.
So today we're diving into a really core subject in biology, one that's, well, absolutely fundamental, especially for those of us prepping for board exams or maybe those critical competitive exams like NEET, UPSC, or even JEE.
Absolutely.
We're talking about life processes, and it's not just about, memorizing facts. It's more about really getting the basic biological operations that keep us and, well, all living things surviving and thriving.
Precisely.
I'm quite keen to start right at the beginning, if that's okay. Can we just get a really clear definition, sort of what are these life processes and what's their scope?
Welcome, and yes, a very warm welcome to all our dedicated learners tuning in. You've really hit on why this topic, life processes, is so critical. Whether you're aiming for NEET or maybe civil services through UPSC, or just building a solid science foundation for any competitive exam, understanding how life works, how it sustains itself is, well, indispensable. So our mission today in this deep dive is to systematically unpack these life processes. We'll go step by step, making sure every concept is clear, that you get that in-depth understanding, and importantly, walk away with an exam-oriented perspective.
Right.
We're going beyond just definitions here. We're really digging into the intricate why and how of these biological things happening inside us, well, right now. #1, what defines life? Unpacking the criteria and molecular movements. Okay, so let's kick off with a question that sounds simple, but it's actually quite profound biologically. How do we, just as observers, tell the difference between something that's alive and something that isn't? Think about what you see every day. You see a dog running, chasing a ball, maybe a cow chewing cud, or even someone shouting across the street. You instinctively know they're living beings, right?
Yeah, definitely.
Their visible actions, their movements. That's often our first clue. But what if that same dog or cow or person was completely still? Maybe sleeping soundly. You still think they're alive, wouldn't you?
Yes, I will.
And how would you know? Probably, you'd look for signs of breathing. That subtle up and down movement, that's a key sign, even without any big activity.
Okay.
Now, plants are a bit trickier. They don't run or shout. Often people think, oh, it's green, so it must be alive.
Right.
But what about plants that aren't green? Or a tree in winter with no leaves? Then the answer usually shifts to, well, they grow over time. That's how we know. Growth. So our first instinct, our common sense, links life with some kind of visible movement. Growth, activity, breathing. But if we look at this strictly, especially for exams, it's not quite enough on its own. Think about a plant that isn't visibly growing right now, like a big mature tree. It looks static, but it's definitely alive.
True.
Or some animals, they can breathe without much visible movement at all, especially when resting. So relying only on visible movement as the single defining feature of life, it's a bit too simple. It doesn't cover everything. That could easily be a trick question, in an objective test.
Okay, so visible movement isn't foolproof.
Exactly. And this leads us to something deeper, more fundamental. Movements that happen on incredibly small scales. Scales we can't see with our naked eyes. We're talking about the movement of molecules.
Molecule.
Yes, constant, intricate molecular movement. Now, the big question for biologists is this invisible molecular movement actually necessary for life? And if you ask the professionals, the answer is a clear yes. In fact, this idea is so central, it's part of that long-running debate. Are viruses really alive?
Ah, yes, the virus question.
Right. Viruses are fascinating because they show no molecular movement on their own outside a living cell. None at all. Only when they infect a host cell do they sort of hijack its machinery and start using its molecules to replicate. This lack of independent molecular movement is a huge reason why there's still scientific controversy about whether they truly count as living or not. And understanding this distinction, this reliance on molecular movement, is really key for your exams.
That's a really profound distinction, tutor. So, okay, if visible movement isn't enough and molecular movement is essential, why? Why are these tiny movements so fundamentally crucial for life? What's the biological reason behind it?
Excellent follow-up. That gets right to the heart of biological organization, something often tested, especially in higher-level questions. Let's think back to earlier classes, maybe class 9 or 10 biology. Living organisms aren't just random piles of stuff, are they?
No, they're structured.
Exactly. Incredibly highly organized structures. There's a clear hierarchy. Simple molecules build complex cells. Cells form tissues. Tissues make organs. Organs work together in systems. It's this intricate, ordered structure that defines life. But here's the catch. This beautiful order is constantly under attack. The environment around us, temperature changes, chemicals, just time passing it all, tends to break down this organization.
Like wear and tear.
Precisely. Think
Okay.
of a well-built house exposed to weather. Without constant upkeep, it starts to fall apart, right? So the consequence is direct. If that internal order breaks down too much, the organism just can't stay alive. It stops functioning as a living thing. Therefore, living creatures must continuously repair and maintain their structures. It's a constant battle against this breakdown, against disorder.
So maintenance is key.
Absolutely. And how is this done? Since everything from tiny parts inside a cell to whole organ systems is ultimately made of molecules, you need constant movement of these molecules for repair and maintenance. It's like a nonstop internal construction crew, always fixing things, replacing parts, keeping everything in working order. This constant molecular activity is the quiet, essential work that keeps everything going. That's a core concept, often tested in descriptive answers.
Wow, okay, that makes sense.
And this understanding leads us perfectly to the next point. These essential continuous repair and maintenance jobs we've just discussed. Well, these are what we collectively call life processes. They're the absolute minimum activities needed just to be alive. So let's define it formally. Life processes are those fundamental biological processes which, together, perform this essential maintenance job in living organisms. The key word here for exams and understanding is essential. They aren't optional extras.
Right. Vital.
And another critical point,
Yeah.
they have to go on continuously, nonstop. Even when an organism looks totally inactive, like right now as you're sitting there listening, or even when you're fast asleep tonight, your body is working away, doing these maintenance tasks, cells repairing, molecules moving. It all requires energy.
Energy. Okay.
Just like maintaining that house needs resources, maintaining biological order needs energy.
Mm-hmm.
To prevent damage, to prevent breakdown, you need energy. And where does this energy come from? It can't just appear out of nowhere inside the body. It has to come from outside the organism.
From the environment.
Yes. So there needs to be a process to transfer this external energy source, what we usually call food, from the outside world into the organism's body. This fundamental process of getting energy and materials from outside. That's nutrition.
Nutrition. Got it.
Now, besides just maintaining things, what if the organism needs to grow? A tiny seed becoming a huge tree or a baby growing up. That requires additional raw material, right?
Mm-hmm. More building blocks.
And again, that has to come from outside. Interestingly, life on Earth is mostly carbon-based. So most food sources are also carbon-based molecules. And depending on how complex these carbon sources are, different organisms have evolved different nutritional processes. A bacterium might absorb simple chemicals, while a tiger hunts complex prey. It varies hugely.
Makes sense.
Okay, so you get these external energy sources inside, but they can be very diverse, chemically speaking. The environment provides all sorts, right? So these diverse sources need to be processed internally. They need to be broken down or sometimes built up into a uniform energy source that the body can use consistently for all that molecular movement, for maintenance, for growth. To do this conversion, you need a whole series of chemical reactions inside the body's cells.
Internal chemistry.
Exactly. And among the most common ways to break down molecules for energy are oxidizing-reducing reactions. Many organisms use oxygen, taken from outside, for these reactions. This whole process, getting oxygen from outside, using it to break down food sources inside cells, and releasing energy, that's what we call respiration.
Respiration. Okay, so that's linked to oxygen and energy release.
Precisely. Now think about scale. A single-celled organism, like an amoeba, his whole surface is touching the environment.
Mm-hmm.
So it doesn't really need complex organs for getting food, exchanging gases, or removing waste. Simple diffusion across its membrane is often enough.
Easy for them, relatively speaking.
Right. But what about multicellular organisms? Like us, or big trees. We get much bigger, much more complex in design. Now, not all cells are touching the outside environment anymore, are they?
No, definitely not. Think about cells deep inside.
Exactly. Cells in your liver, your brain, they're nowhere near the outside air or your food. So simple diffusion just won't cut it. It's way too slow to meet the needs of all those billions of cells.
So diffusion isn't enough for complex life.
Correct. And we learned back in class 9, multicellular organisms developed specialized tissues and organs for different jobs, right?
Yes, specialization.
So it makes sense that taking up food and oxygen also becomes the job of specialized systems. Our digestive system for food, our lungs for oxygen.
Okay.
But this creates a new problem, doesn't it? Food and oxygen come in at one place, mouth, lungs, but they're needed everywhere, by every single cell.
Ah, the distribution challenge.
Precisely.
Yeah.
This creates the need for an efficient internal transportation system. something to carry food, oxygen, hormones, everything, from where they're taken in or made to every other part of the body.
Like a delivery network.
Exactly like that. And finally, think about those chemical reactions for energy. They produce useful energy, yes, but they also create byproducts. Waste.
Stuff the body doesn't need.
Worse than that, stuff that could be harmful, even poisonous if it builds up. These waste byproducts have to be removed. This process of getting rid of metabolic wastes, that's excretion.
Excretion, okay.
And again, in complex organisms, you typically find specialized tissues or organs for excretion, like our kidneys, which means our transportation system also needs to carry these waste materials away from the cells and deliver them to the excretory organs for removal.
It's all connected.
Absolutely. So to summarize, the 4 essential life processes we'll focus on are nutrition, respiration, transportation, and excretion. They all work together like a beautifully coordinated machine to keep life going. Understanding each one and how they link up is crucial for your exams. Right, let's dive deeper into the first one, nutrition. As we said, energy is constantly needed, always. Even when you're just sitting still or sleeping, energy is needed to maintain that order inside us, prevent breakdown.
The maintenance energy.
Exactly. And beyond maintenance, we need materials from outside to grow, develop, make proteins, all sorts of things. This source of both energy and materials is simply food.
Okay.
Now, while everyone needs energy and materials, how they get them varies a lot. This diversity is key for understanding biology. First, we have the autotrophs, think self-feeders. These amazing organisms use simple inorganic materials, mainly carbon dioxide and water, to make their own food.
Like plants.
Green plants are the classic example, yes. And some types of bacteria, too, like cyanobacteria. They are the producers in ecosystems. Then we have the heterotrophs. These organisms can't make their own food from simple stuff. They have to use complex organic substances that other organisms have already made.
So they eat autotrophs or other heterotrophs?
Exactly. And before they can use these complex substances, they need to break them down into simpler molecules. They use biological catalysts, enzymes, to do this job efficiently. So heterotrophs, which includes all animals, fungi, most bacteria ultimately depend on autotrophs for survival, directly or indirectly. The energy flows from the producers.
Got it. Autotrophs make food, heterotrophs consume it. Hashtag 3.1, autotrophic nutrition.
Let's focus on autotrophic nutrition now, especially in green plants, because it's a massive topic for board exams. Their carbon and energy needs are met by photosynthesis.
The big one.
It really is. Photosynthesis is the process where autotrophs take substances from outside CO2 and water and convert them into stored energy, usually carbohydrates like glucose. And this magic happens using sunlight and that green pigment, chlorophyll.
Okay.
The carbohydrates they make are used for their own immediate energy. Needs growth, repair, everything. Any extra carbohydrates not needed right away. They store it. Plants typically store it as starch. It's like their energy savings account.
Starch.
We humans do something similar, actually. We store some excess energy as glycogen in our liver and muscles.
Ah, glycogen. Right.
Now let's break down photosynthesis step by step. It's a sequence of reactions, really important for exam questions. Okay, first, the initial step is absorption of light energy. Chlorophyll, that green pigment, acts like a tiny solar panel, capturing the sunlight.
Step 1, catch the light.
Second, that light energy is then converted into chemical energy. And crucially, at the same time, water molecules, H2O, are split into hydrogen and oxygen. This splitting releases the oxygen we breathe.
Wow, okay, so oxygen is a byproduct here.
It is. And the hydrogen ions and electrons from the water are needed for the next step. Third, this is the reduction of carbon dioxide, CO2, to carbohydrates. Carbon atoms from the CO2 in the air are used to build sugar molecules using the chemical energy captured earlier.
Building the food.
Exactly. Now, an important point. These steps don't always happen instantly, one after the other, in all plants. Desert plants, for example, have a neat trick.
Oh, what do they do?
To save water, they often open their pores, called stomata, and take in CO2 only at night. They make an intermediate compound then. During the day, they use the sunlight energy they capture to finish the process using that stored intermediate.
Clever adaptation.
It really shows how life processes adapt to challenges. Now, if you look at a leaf cross-section under a microscope, you see tiny green dots inside some cells. These are chloroplasts. That's where the chlorophyll is, and that's where photosynthesis happens. Definitely no chloroplasts for diagram questions.
Chloroplasts, got it.
Now, to really prove chlorophyll is needed, let's imagine doing a classic experiment, activity 5.1. You take a potted plant with variegated leaves.
Yeah.
Leaves of green and white patches, like a money plant.
Okay, variegated leaves.
Step 1, keep it in the dark for about 3 days. Why? To make sure it uses up all its stored starch. Start fresh.
De-starch it.
Step 2. Put it in bright sunlight for maybe 6 hours. Let it photosynthesize. Step 3. Pluck a leaf. Carefully trace the green areas on paper so you remember where they were.
Mark the green bits.
Step 4. Boil the leaf in water for a few minutes. This kills the cells, makes the membranes permeable. Step 5. Now put the leaf in alcohol, but heat the alcohol indirectly in a water bath. Okay? Alcohol is flammable. Heat it until the green color dissolves out of the leaf into the alcohol.
Remove the chlorophyll.
Step 6. Take the now pale leaf and dip it in dilute iodine solution. Step 7. Rinse it and look closely. Compare it to your tracing.
And what should happen?
You'll see a blue-black color change, but only in the parts that were originally green. The white parts won't change color. Iodine tests for starch.
Ah, so starch is only made where there was chlorophyll.
Precisely. It's clear proof.
Yeah.
Chlorophyll is essential for photosynthesis for making starch. A classic experiment, very common in exams.
That's incredibly clear, Tutor. We've definitely established chlorophyll's role. But now that brings up another key question for me. How exactly do plants get the carbon dioxide and, the other raw materials they need from the environment for this process?
Excellent question. Very practical and crucial for understanding plant function. Let's look at how they gather these ingredients. The main carbon source, carbon dioxide, primarily enters through those tiny pores we mentioned. Stomata. Usually on the leaf surface, you'll see diagrams of them with their guard cells.
Stomata, okay.
These pores allow massive gas exchange for photosynthesis, though some exchange also happens across stems, roots too. But there's a trade-off. While letting CO2 in, a lot of water vapor can escape through these same pores, especially when it's hot and dry.
Yeah.
That's transpiration. We'll get back to that.
Water loss.
Right. So plants need to control this. They close their stomata when they don't need CO2 for photosynthesis, like at night, or when water is scarce. The opening and closing are controlled by specialized guard cells around each pore.
How do they work?
When water flows into the guard cells, they swell up and bend outwards, opening the pore. If they lose water, they shrink and the pore closes. It's a very neat mechanism, often asked about.
Clever.
And we can prove CO2 is essential with another experiment, activity 5.2. Imagine 2 healthy potted plants, same size.
Okay, 2 plants.
First, dark room for 3 days again. Destarch them. Second, put each on a separate glass plate. Now next to one plant, place a watch glass containing potassium hydroxide, KOH. KOH absorbs CO2 very effectively.
So one plant has CO2 removed from its air.
Exactly. The other plant is the control, no KOH. Third, cover both plants with separate bell jars, sealing the bottom to the glass plate with Vaseline. Make it airtight.
Airtight setups?
Fourth, put both in sunlight for a couple hours. Finally, pluck a leaf from each and test for starch with iodine.
And the result?
You'd expect starch, blue-black color, only in the leaf from the plant without the KOH. The one where CO2 was removed won't show much starch, if any.
Proving CO2 is needed.
Absolutely.
Yeah.
A very standard experimental setup question. Now, besides CO2, plants need other things. Water, obviously, used in photosynthesis itself and for transport. Terrestrial plants get water from the soil via their roots.
Water from roots.
And minerals too, nitrogen, phosphorus, iron, magnesium, all absorbed from the soil by the roots. Nitrogen is super important for making proteins and DNA. It's usually taken up as nitrates or nitrites from the soil.
Minerals from soil, too.
Or sometimes, especially in legumes like peas or beans, they get nitrogen through a partnership with bacteria that can take nitrogen gas from the air and fix it into usable compounds in the soil. It's all about gathering those essential raw materials. Hashtag tag 3.2 heterotrophic nutrition. Okay, moving on from the self-feeders, let's talk heterotrophic nutrition. Remember, these are the organisms that can't make their own food. They get complex organic stuff from other organisms and break it down using enzymes.
They depend on others.
Directly or indirectly, yes. Animals, fungi, they're all heterotrophs. And how they get their food varies enormously. Think about a cow eating grass versus a lion hunting a deer.
Very different strategies.
Totally different. Based on food type, stationary versus mobile, availability, how they get it, leads to different adaptations. We see a few main strategies. Some break down food outside their body first, then absorb the simple nutrients. Fungi do this, bread molds, mushrooms.
Yeah.
They release enzymes onto dead stuff, digest it externally, then absorb. This is saprophytic nutrition.
Saprophytic, like decomposers.
Often, yes. Then you have organisms, like most animals, that take in whole food material and break it down inside their bodies. Ingest, digest, absorb, etc. That's holozoic nutrition.
Holozoic, like us.
Exactly. And a third major type is parasitic nutrition. These organisms live on or inside another living organism, the host, and get nutrition from it without killing it immediately.
Parasites.
Right. Like Cuscuta, that yellow vine on plants, or ticks, lice, tapeworms in animals. They've evolved ways to siphon off nutrients from their living host. All important types to know for classification questions. So given all this variety in food and feeding styles, it makes sense that digestive systems vary too, right? Simpler organism, simpler system. Take single-celled organisms. Their whole surface touches the environment. Food intake is often quite direct. Look at amoeba. It uses these temporary false feet pseudopodia to slow around and engulf a food particle.
Wraps around it.
Exactly. Forms a little bubble inside called a food vacuole. Enzymes go into the vacuole, break down the food, nutrients diffuse out into the cytoplasm. Undigested stuff just gets pushed out.
Simple but effective.
Then you have paramecium, also single-celled, but it has a fixed shape, like a slipper, and it takes food at a specific spot, like a tiny mouth. It uses hair-like cilia all over its surface to beat and sweep food particles towards that spot.
More organized, even in a single cell.
It shows that even simple life adapts. But now let's move to the really complex systems. Hashtag tag 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 3.4, nutrition in human beings.
Okay, let's tackle nutrition in human beings. Our system is basically a long tube, the alimentary canal, running from mouth to anus. But it's not just a pipe. Different parts are highly specialized. Let's trace the food's journey. This is prime territory for long answer questions and diagrams.
Okay, starting at the beginning.
The mouth. First stop. Food needs breaking down mechanically. That's the job of our teeth-crushing, grinding, making it smaller, more uniform.
Chewing.
And it needs wetting, right, to slide down smoothly. That's where saliva comes in from our salivary glands. You know when your mouth waters?
That's saliva. And it's not just water. It contains a crucial enzyme, salivary amylase. This enzyme starts breaking down complex starch from things like bread, rice, potatoes, into simpler sugars like maltose.
So digestion starts right in the mouth.
Chemical digestion of starch starts immediately, yes. And our muscular tongue mixes the food with saliva, helps chewing, forms a soft ball called a bolus, ready for swallowing. Remember activity 5.3. If you take starch solution in 2 tubes, add saliva to 1A, but not the other B, wait a bit, then add iodine.
Tube B turns blue-black, starch present, but 2A doesn't.
Exactly, because the salivary amylase in tube A broke down the starch. Simple proof of enzyme action, very common practical question. Okay, food is chewed, mixed with saliva. Now it needs to move down. This isn't just gravity. There are rhythmic muscle contractions all along the gut, pushing the food. These waves are called peristalsis.
Peristalsis, like squeezing a tube.
Good analogy. It moves food from the mouth down the esophagus, food pipe, which connects the throat to the next major stop, the stomach. A J-shaped muscular bag. It expands to hold food, acts like a churn. Its muscular walls contract, mixing food thoroughly with gastric juices secreted by gastric glands in the stomach wall. These glands release 3 key things you must know. First, hydrochloric acid, HCl, makes the stomach very acidic, pH 1.5, 3.5. This helps activate an enzyme and also kills most ingested bacteria, like a sterilizing wash.
Kills germs, helps digestion.
Second, pepsin. This is a protein-digesting enzyme and needs that acidic environment created by HCl to work properly. It starts breaking down large protein molecules into smaller pieces called peptides. Protein digestion begins here.
Pepsin for proteins.
Third, mucus, a thick, slimy layer that coats the stomach lining. Crucial. It protects the stomach wall from being digested by the strong acid and pepsin.
Protection. So that's why acidity or ulcers can be a problem if that balance is off.
Precisely. It's often related to issues with acid production or the protective mucus layer. Now, food doesn't just dump out of the stomach. Its exit is controlled by a ring of muscle, a sphincter, at the bottom. It releases the partially digested food, now called chyme, into the small intestine bit by bit.
Controlled release.
Very controlled. Into the small intestine. This is where the main event happens. It's incredibly long, maybe 6 to 7 meters, coiled up tightly in the abdomen.
That long. Wow.
And its length varies. Herbivores eating tough plants need longer ones than carnivores eating easily digestible meat. Here, digestion of carbs, proteins, and fats is completed.
The main site of digestion and absorption.
Absolutely. The small intestine receives help from 2 crucial accessory organs. First, the liver, our largest internal organ. It produces bile juice, which is stored in the gallbladder and released into the small intestine. Bile has 2 main jobs. 1, it makes the acidic chyme from the stomach alkaline because the enzymes working here need an alkaline environment. 2, it deals with fats.
How does it deal with fats?
Fats arrive as large globules. Bile salts break these large globules into tiny droplets. It's called emulsification. This hugely increases the surface area for fat-digesting enzymes to work on. Think of how soap breaks up grease.
Emulsification increases surface area.
Our second accessory organ, the pancreas. It releases pancreatic juice into the small intestine. This contains powerful enzymes. Trypsin continues protein digestion, and lipase breaks down those emulsified fats. Pancreatic amylase handles carbohydrates too.
Liver gives bile, pancreas gives enzymes.
And the walls of the small intestine itself also secrete intestinal juice with more enzymes. These finish the job completely. Proteins, amino acids, complex carbs, glucose, fatty acids, and glycerol. Now they're small enough to be absorbed.
Final breakdown complete.
Now for absorption in the small intestine. The inner lining isn't smooth. It's covered in millions of tiny, finger-like projections called villi. These villi vastly increase the surface area for absorption.
The more surface area means faster absorption.
Much faster, much more efficient. Each villus has a rich network of blood capillaries. The digested nutrients, glucose, amino acids, fatty acids, glycerol, pass through the intestinal wall into these capillaries. The blood then transports these nutrients to every cell in the body. Cells use them for energy, building new tissues, repairing old ones. Incredible efficiency.
Amazing system. What happens to the leftovers?
The unabsorbed stuff, mostly fiber and water, passes into the large intestine. Its main job is to absorb most of the remaining water, making the waste more solid.
Water absorption.
Right. The final waste material, feces, is stored temporarily in the rectum and then eliminated from the body through the anus, controlled by the anal sphincter. That completes the journey. Knowing each part and its function is key for exams.
Definitely a lot to remember there.
And just quickly, that more to know on dental caries. Tooth decay. Bacteria in your mouth feed on sugars, produce acid. Acid dissolves tooth enamel. Bacteria plus food particles yield plaque. Plaque traps acid against the tooth. Brushing removes plaque. Simple. If untreated, it can reach the pulp, cause infection, pain. So brush regularly. Okay, so we've got the food digested and absorbed. Now, how do cells actually use it to get energy for all those life processes? That's respiration. Different organisms do it differently, but the very first step is almost universal. Breaking down glucose, 6-carbon sugar, into a 3-carbon molecule called pyruvate. This initial split happens in the cytoplasm of the cell. Doesn't need oxygen.
Glucose to pyruvate in the cytoplasm. Always the first step.
Yeah, pretty much always the first step in glucose breakdown for energy. What happens next to pyruvate depends crucially on whether oxygen is available or not. This leads to different pathways. Let's go step by step. This is classic exam material. Hashtag 4.1 pathways of glucose breakdown. Path 1, anaerobic respiration. This means without oxygen. A good example is fermentation in yeast. Here, pyruvate is converted into ethanol, a 2-carbon alcohol, and carbon dioxide. Think brewing, baking bread.
Yeast fermentation, ethanol and CO2, no oxygen.
Exactly. You can show the CO2 production with activity 5.5. Put yeast and sugar solution in a tube, connect it via bit tube to lime water. The lime water turns milky because the yeast produces CO2. This anaerobic process releases some energy, but much less than with oxygen. Only about 2 ATP per glucose.
Less efficient.
Much less. Path 2, aerobic respiration. This happens with oxygen. It's the main pathway for us and most complex organisms. After pyruvate forms in the cytoplasm, it moves into the mitochondria, the cell's powerhouse.
Mitochondria, right. Yeah.
Inside the mitochondria, with plenty of oxygen, pyruvate is completely broken down. The end products are carbon dioxide and water. And crucially, this releases a lot more energy. Way more efficient. Around 36-38 ATP per glucose.
Aerobic. In mitochondria, needs oxygen, yields lots of energy, ATP.
Perfect summary. Path 3, lactic acid fermentation. This is an interesting one. It happens when there's a lack of oxygen, even in aerobic organisms, like in our muscle cells during really intense exercise.
Like sprinting.
Exactly. Your muscles need energy fast, maybe faster than oxygen can be supplied. So as an emergency measure, pyruvate gets converted into lactic acid, another 3-carbon molecule.
Lactic acid, isn't that what causes muscle cramps?
It is. That buildup of lactic acid contributes to muscle fatigue and soreness. Once you rest and oxygen supply catches up, the lactic acid is usually cleared.
So 3 fates for pyruvate depending on oxygen. Anaerobic, ethanol CO2 or lactic acid, low energy, or aerobic, CO2 water, high energy.
You've got it. Diagrams showing these pathways are very common in textbooks and exams. Make sure you understand them well.
So these processes release energy, but how is that energy actually captured and then used by the cells for things like muscle contraction or building proteins? Is there like a specific molecule for that?
Absolutely great question. It's not just released randomly as heat. Most of the energy released during respiration is immediately used to make a special high-energy molecule called ATP adenosine triphosphate.
ATP, the energy currency.
Exactly. Think of it as the cell's universal rechargeable energy battery or currency. It's made by taking ADP, adenosine diphosphate, and adding a phosphate group, pi, using the energy from glucose breakdown. So ADP plus pi plus energy ATP.
Charging the battery.
Precisely. Then, whenever the cell needs energy for anything, muscle contraction, sending nerve signals, building molecules, pumping things across membranes, these are called endothermic reactions needing energy input, it spends ATP.
How does it spend it?
It breaks off that third phosphate group, ADP plus pi. That breakage releases a nice, usable packet of energy, about 30.5 kilojoule. This released energy directly powers the cellular activity.
So ATP stores energy, and breaking it releases energy where needed.
Exactly. It's an incredibly efficient way to manage energy flow within the cell. Central concept for cellular biology. Hashtag 4.2 gas exchange in organisms. Okay, so aerobic respiration needs a steady supply of oxygen and produces carbon dioxide that needs removing. This brings us to gas exchange. How do organisms swap these gases with their environment? Start with plants. They primarily use stomata, those leaf pores again. They also have air spaces between cells inside, so gases can diffuse around. CO2 and O2 move by diffusion, high concentration to low concentration.
Simple diffusion, mostly.
Yes, and the direction depends on what the plant is doing. At night, no photosynthesis, only respiration. So they take in O2, release CO2 during the day. They're photosynthesizing rapidly. They use up the CO2 from respiration and take in more CO2 from the air. The main gas released during the day is oxygen from photosynthesis.
So the net gas flow changes between day and night.
It does. Very dynamic. Now, animals. They've evolved specialized organs for gas exchange, and it differs a lot between water dwellers and land dwellers.
Aquatic versus terrestrial.
Right, aquatic organisms, like fish, they need oxygen dissolved in water. But there's much less oxygen dissolved in water than in air.
So it's harder for them?
That means they have to work harder, breathe faster. Watch a fish, activity 5.6. They constantly gulp water, forcing it over their gills. Gills have a huge surface area, packed with blood vessels, to grab that scarce dissolved oxygen efficiently. Their breathing rate is much higher than ours.
Faster breathing to compensate for low oxygen levels.
Exactly. Terrestrial organisms use oxygen from the air, which is much more abundant. Their respiratory organs, like our lungs, are designed for a large surface area in contact with air. But this surface needs to be thin and delicate for diffusion, so it's usually internal, protected inside the body, which means you need passages to get air in and out.
Tubes leading to the exchange surface.
Right, and you need a mechanism to actively move air in and out, ventilation or breathing.
That makes perfect sense, Tutor. The adaptations are fascinating. So how does this elaborate system apply to us humans? What's the specific structure of our respiratory system, and how does it manage this vital gas exchange so efficiently?
Excellent question. Let's zoom in on the human respiratory system. A really well-designed system and a major exam topic. Air enters through the nostrils and gets filtered by hairs, warmed and humidified by mucus lining the nasal passages.
Filtered and prepared.
Then it goes down through the pharynx, throat, larynx, voice box, into the trachea, windpipe. The trachea and the larger tubes branching off it, bronchi, have rings of cartilage. Why?
To keep them open, prevent collapsing.
Exactly. Ensures the airway stays open. The trachea branches into two main bronchi, one for each lung. These then branch again and again, like a tree, into smaller bronchioles. These tiny tubes end in clusters of microscopic air sacs, the alveoli, millions of them. These are the main sites of gas exchange.
Alveoli are where the action happens.
Precisely. Their walls are incredibly thin, just one cell thick, and they're covered in a dense network of tiny blood capillaries. This maximizes surface area and minimizes diffusion distance. Super efficient design.
Okay, how does the air actually move in and out, the breathing part?
The breathing mechanism. When you inhale, your rib muscles contract, lifting the ribs up and out. Your diaphragm muscle below the lungs contracts and flattens. This increases the volume of your chest cavity.
Just expands.
Right. Pressure inside drops. Air rushes in to fill the lungs and alveoli. Then, gas exchange at the alveoli. Blood arriving is low in oxygen, high in CO2. Oxygen diffuses from the high concentration in alveolar air into the blood. CO2 diffuses from the high concentration in the blood into the alveolar air to be breathed out.
Simple diffusion across the thin walls.
Driven by concentration gradients, yes. When you exhale, rib muscles relax, ribs move down and in. Diaphragm relaxes, moves up. Chest volume decreases, pressure increases, air is pushed out.
Inhale active, exhale passive mostly.
Normal exhalation is largely passive, yes, though you can force air out actively too. Interestingly, your lungs don't fully empty when you exhale. There's always some air left, the residual volume. This ensures gas exchange can continue even between breaths and keeps the alveoli from collapsing.
Residual volume. Smart.
And here's that mind-blowing fact. The total surface area of all your alveoli, about 80 square meters, like a tennis court, packed inside your chest. That huge area is why gas exchange is so fast and efficient.
Incredible.
Now, for big animals like us, diffusion alone is way too slow to get oxygen everywhere. Remember the 3 years to the toes idea?
Yeah, so how do we speed it up?
Respiratory pigments, molecules that bind oxygen strongly in the lungs and release it where needed in the tissues. Our pigment is hemoglobin, found in red blood cells. It grabs oxygen efficiently and carries it rapidly via the blood.
Hemoglobin in RBCs does the delivery job.
Exactly.
Yeah?
CO2 is mostly transported, dissolved in blood plasma, as it's more soluble than oxygen. And that crucial health node, smoking. It damages the tiny cilia, the little hairs lining your airways that sweep out dust and germs. Damage the cilia, and all that harmful stuff in smoke gets into your lungs, causing infections, coughs, and drastically increasing cancer risk. Plus, oral cancer from chewing tobacco, it's just not worth it. Say no. Right, so we've got food in, energy released, oxygen delivered. But how does it all get around? We need transportation, the body's delivery network, and the main player here is blood.
Blood carries everything.
Pretty much. Food, oxygen, hormones, waste. We know from class 9 it's a fluid connective tissue. Plasma is the liquid part, carrying dissolved stuff in cells. Red blood cells, RDCs, carry oxygen using hemoglobin. White blood cells fight infection. Platelets clot blood.
Plasma, RBCs, WBCs, platelets.
To make this work in a complex body, you need 3 things: a pump, heart, a network of tubes, blood vessels, and a repair system, platelets. Hashtag 5.1, transportation in human beings. Let's focus on transportation in humans, our cardiovascular system. Hugely important exam topic. The pump is the heart, muscular organ about fist-sized. Crucially, it has 4 chambers. Why 4?
To keep oxygenated and deoxygenated blood separate.
Exactly. Complete separation. This is vital for efficiency. CO2-rich blood needs to go to the lungs. O2-rich blood from the lungs needs to go to the body. No mixing allowed for maximum oxygen delivery. Let's trace the path. Double circulation. This is key. Follow along, perhaps visualizing a diagram. Path 1, pulmonary circuit to lungs and back. Deoxygenated blood from the body enters the top right chamber, the right atrium. It relaxes to receive blood. Then the right atrium contracts, pushing blood into the bottom right chamber, the right ventricle, which relaxes to fill. Then the right ventricle contracts, pumping this deoxygenated blood to the lungs via pulmonary artery.
Right side pumps blood to the lungs, deoxygenated.
Perfect. Path 2, systemic circuit to body and back. Oxygenated blood returns from the lungs, via pulmonary veins, to the top left chamber, the left atrium. It relaxes to receive blood. Left atrium contracts, pushes blood into the bottom left chamber, the left ventricle, which relaxes to fill. Now, the left ventricle is the powerhouse. Its walls are the thickest. It contracts forcefully, pumping oxygenated blood out to the entire body via aorta.
Left side pumps blood to the body, oxygenated. Stronger pump.
Exactly. Ventricle walls are thicker than atrial walls because they pump blood much further. And the left ventricle is thickest because it pumps to the whole body against higher pressure. And between chambers and where blood leaves the ventricles, there are valves, one-way doors. They ensure blood flows only forward, preventing backflow. Crucial for efficiency.
Valves ensure one-way flow.
This 4-chambered heart with complete separation allows highly efficient oxygen supply. Vital for warm-blooded animals like birds and mammals who need lots of energy to maintain body temperature.
So it's linked to being warm-blooded.
Strongly linked. Contrast with amphibians and most reptiles, they often have 3-chambered hearts, tolerate some blood mixing, are cold-blooded, body temp varies with environment. Fish have only 2 chambers. Heart pumps blood to gills, then it goes straight to the body. That's single circulation.
We have a double circulation because blood goes through the heart twice per circuit. Once for lungs, once for body.
Precisely. That's the definition of double circulation. Important distinction. Now, the network of tubes, blood vessels, 3 main types, arteries, carry blood away from the heart under high pressure. So they have thick, elastic, muscular walls.
Arteries, away, thick walls, high pressure.
Veins carry blood back towards the heart, lower pressure now. Walls are thinner. They often have valves, especially in the limbs, to prevent backflow against gravity.
Veins towards thin walls, low pressure, valves.
They're capillaries. The tiny microscopic vessels connecting arteries and veins within tissues. Walls are super thin, 1 cell thick. This is where the exchange happens. O2 and nutrients out to cells, CO2 and waste in from cells.
Capillaries exchange site, very thin walls.
Right. Arteries branch into capillaries. Capillaries join up to form veins. What if there's a leak, injury, bleeding? We need to stop blood loss and maintain pressure. That's the job of platelets. They circulate, find the leak, and clump together, helping form a blood clot to plug the hole. Essential maintenance.
Platelets for clotting.
And there's another fluid, lymph or tissue fluid. Some plasma and white blood cells leak out of capillaries into the spaces between cells. This fluid bathes the cells.
Lymph.
Fluid around the cells. Yes. It collects in lymphatic capillaries, then larger lymph vessels, eventually returning to the bloodstream near the heart. Lymph helps transport digested fats from the intestine and drains excess fluid from tissues, preventing swelling.
So it's like a secondary circulation system?
In a way, yes, focused on fluid balance and fat transport, and also immune functions. Quick note on blood pressure. Force of blood on vessel walls, higher in arteries, measured with a sphygmomanometer. Normal is around 120, systolic ventricle contracting over 80, diastolic ventricle relaxing millimeter Hg. High blood pressure, hypertension is dangerous, increases risk of stroke, heart attack.
20/80 is the benchmark.
Roughly, yes. And remember that diffusion fact. 3 years for oxygen to toes shows why hemoglobin and rapid circulation are vital. You can even check hemoglobin ranges, activity 5.7, compare humans, animals, males, females. It varies based on needs. Hashtag check 5.2, transportation and plants.
Okay, let's switch gears to transportation and plants. They're different, don't move much, lower energy needs, lots of dead tissue, wood, so their transport can be slower.
But distances can be huge, right? Tall trees.
Exactly, that's the challenge. Raw materials, water, minerals, come from soil via roots. Energy, sugars, comes from leaves via photosynthesis. Diffusion is too slow for these long distances.
So they need pipes, too.
They do. Two main types of vascular tissue, xylem and phloem. Hashtag 5.2.1 transport of water via xylem. Xylem transports water and dissolved minerals. It's made of vessels and tracheids, basically. Long, hollow, dead cells joined end to end, forming continuous pipes from root to leaf.
Xylem for water, upwards. Primarily upwards, yes. Driven by two main forces. One, root pressure, especially at night. Root cells actively pump minerals in from the soil. Water follows by osmosis. This creates a positive pressure that pushes water up the xylem a bit.
A push from the bottom.
2. Transpiration pull. This is the main driver, especially during the day. Water evaporates from leaves through stomata, transpiration. This creates a tension, or suction, that pulls water up the continuous xylem column, all the way from the roots, like sucking water up a straw.
A pull from the top, driven by evaporation.
Exactly. You could show transpiration with activity 5.8. Cover a plant and its pot with soil cover with a plastic bag. Put it in sun. Water droplets condense inside the bag, proving water loss from the plant.
Simple proof.
Transpiration isn't just water loss. It helps pull water and minerals up and cools the plant. So root pressure gives a push. Transpiration gives a powerful pull. That's how water gets to the top of even the tallest trees. Hashtag 5.2.2, transport of food and other substances via phloem. Now for food transport, phloem. This process is called translocation. It moves the soluble products of photosynthesis, mainly sucrose, from where they're made, leaves the source, to where they're needed or stored. Roots, fruits, growing tips, the sinks.
Phloem for sugars, source to sink.
Right. Phloem also carries amino acids, hormones. It uses living cells called sieve tubes, helped by companion cells. Unlike xylem, mostly up, phloem transport can go up or down.
Bidirectional.
Yes, depending on the plant's needs. And here's a key difference. Translocation in phloem requires energy, ATP. It's an active process.
Xylem is passive pull, phloem is active push movement.
Broadly speaking, yes. Sugars are actively loaded into the phloem at the source using ATP. This draws water in by osmosis, creating high pressure. This pressure pushes the sugary sap along the phloem towards the sink, where sugars are unloaded, also often requiring energy.
So it uses energy to create pressure flow.
Exactly. This allows the plant to direct food where needed, like sending stored sugar up to growing buds in spring. Very adaptable. Okay, final life process. Excretion, we've got nutrients, energy, transport, but metabolism creates waste products, especially nitrogenous waste from breaking down proteins that are harmful and need removing.
Getting rid of the metabolic garbage.
Precisely. That's excretion. Strategies vary. Unicellulars often just diffuse waste out. Multicellulars need specialized organs. Hashtag 6.1 excretion in human beings. The human excretory system. Key components. Pair of kidneys, pair of ureters, tubes from kidneys, a urinary bladder, storage bag, and a urethra, tube for exit.
Kidneys, ureters, bladder, urethra, got it.
Kidneys are the filters, located in the abdomen, either side of the spine. They filter waste from blood to produce urine. Urine travels down ureters to the bladder for storage, then exits via urethra.
So how do kidneys actually make urine?
Purpose is to filter blood, remove nitrogenous waste like urea and uric acid. The basic filtering unit in the kidney is the nephron. Millions per kidney.
Nephrons do the filtering.
Each nephron has two parts, a tangled ball of capillaries called the glomerulus, and a long coiled tubule starting with a cup called Bowman's capsule surrounding the glomerulus. Two main steps happen in the nephron. One, filtration. High pressure in the glomerulus forces water, salts, glucose, amino acids, ureas, basically small stuff out of the blood and into Bowman's capsule, like forcing liquid through a sieve. Large things like proteins and cells stay in the blood. This filtered liquid is called filtrate.
Step 1, sieve the blood, filtration.
2. Selective reabsorption. The filtrate flows down the long tubule. Now the body needs to take back the useful stuff. Glucose, amino acids, most salts, and most of the water are actively transported back out of the tubule and into the blood capillaries surrounding it.
Step 2, take back the good stuff, reabsorption.
Exactly. It's highly selective. The amount of water reabsorbed depends on how hydrated you are. We filter maybe 180 liters a day, but only excrete 1 to 2 liters of urine. Shows how much gets reabsorbed. The remaining fluid, now concentrated waste, mostly urea, and excess salts dissolved in water, is urine.
Incredible efficiency.
Urine collects, goes down ureters to the bladder. Bladder stretches, signals the brain, and eventually urine is released via the urethra. The bladder has nervous control, so we learn to control urination. And that more to know on artificial kidney. Hemodialysis. If kidneys fail, wastes build up, which is fatal. Dialysis uses a machine to clean the blood.
How does that work?
Patient's blood flows through tubes with semi-permeable membranes bathed in a special dialyzing fluid. Waste diffuses out of the blood into the fluid, because there's none in the fluid. Clean blood goes back to the patient. It filters, but importantly, it doesn't do selective reabsorption like a real kidney. Hashtag 6.2 excretion implants. Finally, excretion in plants. Very different from animals. No specialized organs like kidneys.
How do they manage waste then?
Gaseous waste, O2 from photosynthesis, CO2 from respiration, is handled via stomata. Excess water via transpiration. Other wastes, they have clever tricks. Stored in cellular vacuoles. Stored in leaves that fall off. A convenient way to dump waste seasonally. Stored as resins in gums in old non-functional xylem wood. Some can even excrete waste into the soil around their roots.
So they store it, dump it with falling parts, or release it slowly.
Pretty much. Different strategies for a different lifestyle. Hashtag out, check out. And that brings us to the end of our deep dive into life processes. We've covered how life hinges on molecular movement and maintenance. We explored nutrition autotrophs making food, heterotrophs consuming it, and the detailed journey through the human gut. Then respiration breaking food for ATP energy, aerobic versus anaerobic pathways, and gas exchange in plants and humans. Followed by transportation, the human circulatory system's double loop, heart, vessels, blood, lymph, and plant transport via xylem and phloem.
Right.
And finally, excretion-removing metabolic wastes via human kidneys and nephrons and the various strategies plants use. The big takeaway really for exams and just understanding biology is how interconnected these all are. They work together seamlessly. Mastering these concepts, not just memorizing, is absolutely key for NEET, UPSC boards, you name it.
I truly feel I have a much clearer and deeper understanding of these concepts now, Tutor. It really helps to connect all these intricate systems. Thinking about the complexity we've discussed, what's a final big picture idea for us students to keep in mind? Perhaps something that goes beyond just memorizing the processes for an exam.
That's a great thought to end on. My advice is always focus on the how and why, not just the what. Visualize the processes. How does blood flow? How does ATP release energy? Connect it to your own body. That makes it stick. Remember, this topic is a favorite for examiners. So revise thoroughly. Focus on definitions, yes, but also diagrams, mechanisms, the logic behind it all.
Practice questions, too.
Definitely. Practice past year questions on life processes. See where you stand, what needs more work. And seriously, if doubts remain, don't hesitate. Ask, clarify. Keep exploring, questioning, learning. And maybe a final thought. As we see how complex and vital these systems are, circulation, respiration, excretion, it really highlights how precious life is, how finely balanced these processes are. Makes you appreciate the life processes on a whole new level, doesn't it? Something to think about.